And so here we start off at proerythroblasts, and then we end up with erythroblasts, which has 3 divisions, but we don't care about these. We really just want to know the term erythroblasts. And it's already designating what it's going to turn into erythro. And remember that blasts were always builder. They were always an immature form of the cell, right? The blast will eventually turn into a cyte in some way or another. And so erythroblasts will turn into erythrocytes. And then once our erythroblast stage is over, the nucleus will be ejected from the cell. And we end up with something we refer to as a reticulocyte, which is just simply an immature red blood cell. An immature red blood cell. And it takes approximately 24 hours for once it's like swimming around with all the other red blood cells to mature into a mature red blood cell, as we've learned about last week, with its concave shape, its no nucleus, its hemoglobin molecules, carrying around oxygen. And now where things are getting new is that remember that the lifespan of a red blood cell was about 120 days, roughly 3 months. And what will happen is that these red blood cells, when they've reached their full lifespan, you will have cells such as macrophages. Remember we talked about macrophages? They come from monocytes. And these are white blood cells. They will find and break down red blood cells that are old or damaged, and they will do 2 things. They will grab that heme group and recall, I'm going to go ahead and just switch back just so we can get a nice little refresher on it. And the heme group, remember that when we talked about red blood cells, inside of them there is a protein that carries around oxygen, and it relies on these heme groups, which, when you look at it at a molecular level, is that it's an iron-based molecule that binds to oxygen, because oxygen and iron do have a very good relationship together. They bind really well together. And so that same heme group is going to be recycled, broken down, and that iron and all the amino acids that made up this protein, that made up these beta chains, alpha chains, all those amino acids will all be recycled and transported by a protein called transferrin. In the bloodstream, you have proteins that are specialized, or their only purpose is to just transport molecules. And in this case, transferrin, if something's swimming around in plasma, grabs iron, and it will send it right back to the red bone marrow, so that all those components, the amino acid and the iron, can be replaced, or they can be used to replace dead or old red blood cells. And that's the role of macrophages. But there is one more pathway that these macrophages you can follow along with. And that's that when other components of the actual cell are broken down, it generates biliverdin, which is this green substance. It's going to sound something like bile, which we'll learn about in digestion, biliverdin, and then this will be converted into bilirubin, which will be something that will be much more common in a lot of your medical pathways. And so this bilirubin will go into the bloodstream, right? The opposite was heme and iron and amino acids, everything returning to the bone marrow, but some stuff needs to be removed. Some stuff from this red blood cell cannot be used, and it is a waste. And so biliverdin will be converted into something we refer to as bilirubin. And then we're going to see albumin. Recall that on our previous slide deck, we saw that one of the components of plasma was albumins. And so we're seeing another function of our plasma protein, albumin. And in this case, it carries around bilirubin in the bloodstream, very similar to the way that transferrin was carrying around iron. So these are just transportation molecules in a sense. They can bind to iron, they transport it. This one, albumin, binds to bilirubin and transports it. It'll arrive at the liver, and this bilirubin will eventually be excreted into bile. And this, when we look at our liver now, our liver plays a big role in digestion in that it produces something called bile, this green substance that helps break down fats. But this stuff sometimes in excess, when you need to remove it, because it's just sometimes you don't need as much bilirubin like to make bile, it will be excreted into your large intestine, which stores feces, and that same bilirubin will be consumed and used up by bacteria in the large intestine. and they will convert bilirubin into urobilins or stercobilins. And then this will be eliminated in your feces. And this is what gives feces that brown, yellowish color. It comes from bilirubin being consumed by bacteria. So you can see bilirubin comes from red blood cells, goes to the liver, and then goes to the large intestine and is removed through feces. So even the liver is responsible for removing things that from the red blood cell that we don't need anymore, the waste. Something to clarify here is when we have amino acids, some can be salvaged. When you look at, say, this protein, this is a protein made up of amino acids, and this was our heme group. And our heme group will turn into biliverdin, or it'll be recycled as heme if you don't have enough. And amino acids can come off of this, and the macrophages can recycle those amino acids. But in instances where we don't need those amino acids, you can see that those same amino acids that come from the breakdown of red blood cells will stay in the bloodstream and then go to the kidneys, and then the kidneys will remove that same hemoglobin proteins through urination. So you can see that feces and urine gets rid of components of red blood cells after that 3-month cycle. And so let's go through these bodies of text here. When we're forming red blood cells, just that segment that we saw here in event 1, this is considered erythropoiesis. This is the formation of red blood cells. Myeloid tissue is the origin of all those red blood cells. And you find red bone marrow in the ribs, the vertebra, sternum, skull, the scapula, the pelvis. And you don't need to know these examples, but you should know that erythropoiesis occurs in red bone marrow, and the original tissue, stem cell population, is myeloid tissue. Fat or yellow marrow was always fat molecules, as you've learned about adipose tissue and its adipocytes, but yellow marrow can be turned into red marrow. In other words, those myeloid tissues take over that space, utilizing the energy from those fat molecules. And so what's the point of learning this little box here? It's sort of like a stage of developments when we learn erythroblast versus reticulocyte versus mature red blood cells. And so erythroblasts begin producing hemoglobin, the main carrying molecule that carries oxygen in red blood cells. Normal blast, it's kind of like saying this blast is looking more normal, looking more like a normal red blood cell because it begins to lose its nuclei. That's a nice way of remembering that. And once it loses its nuclei, it becomes a reticulocyte. And so only 80% of your red blood cells at any given time are mature. And after that, about 20%, after 2 days, they're converting into mature red blood cells. These macrophages, as we're going to see in lymph system, is that they monitor things that are flowing through the fluid in general throughout your entire body, which gets a little more complicated. It's not just plasma, but they are monitoring anything that's going through, and so they engulf through a process called phagocytosis. You may have heard of this in microbiology. And also hemolyze means to break apart, right? To break something apart, especially our heme groups. And here, transferrin is a plasma protein. We don't see it on our list of plasma proteins, but it is a protein that flows, and its job is just to recycle that iron. And that heme group will either be recycled, the iron that's making it up, and every other bit of the components of it will turn into biliverdin or bilirubin, and then bilirubin, enter the bloodstream and be carried around by albumin, which is missing from this text here. Albumin, to the liver, and then the liver will put it in the large intestine where it will be removed through feces. And anything that can, any hemoglobin, any of these molecules that cannot be broken here, cannot be broken down, because this is just the heme group. Recall that this whole object is hemoglobin, the entire 4 subunits and their 4 heme groups. The heme group is only this iron-carrying centers, the 4 that you're seeing there. And so in this context, hemoglobin, the meaning the entire molecule that is not used up, will be removed through urine. So if it cannot be broken down, it will be excreted in urine. But if it's broken down, for the most part, it will be removed through the large intestine. So that's the big difference. That as we look at this pathway, you can go in 3 directions. One's not so obvious. One, as we go down here, you can see that any hemoglobin that's not going to be phagocytized or broken apart will just be excreted through urine. Otherwise, your macrophages will be responsible for breaking down and recycling the iron in those heme groups, and anything that cannot be recycled will again take this biliverdin pathway to the liver, to the large intestines. And again, urobilins just means any bilirubin that has been affected or consumed by bacteria. You can even write here, bacteria consumed. Put bacteria utilize. And something that we haven't really discussed in large, like in digestive at all in general, but I think we have a general idea that the part in the small intestine and the large intestine, their role is to grab anything going through the circular tube that is your small intestine. As food is going through there, things are being absorbed into blood vessels and being transported throughout. Your large intestine, even though all the nutrients have already probably been absorbed in the small intestine, when things enter the large intestine, they already resemble feces. It's already utilized all the food that you had and already broken it, like taking all its nutritional value for the most part. And the large intestine works as 2 things, a storage pouch for feces, and also it can also reabsorb things. But it doesn't care about absorbing nutrients like proteins, nucleic acids, lipids, and carbohydrates. What it cares about is absorbing typically water and ions such as calcium, magnesium, potassium, sodium, and it grabs it out of all that feces, grabs all that water and any excess ions that it needs, it grabs it and places it right back into blood vessels. So your body can eliminate these urobilins by feces, or sometimes they actually go into circulation, into the blood vessels, and then they're excreted through the kidneys. Because remember that the kidneys, what they do is that they are removing waste from your blood vessels. From your blood vessels. And occasionally, if you have excessive amount of red blood cells breaking down, you can yield red or brown urine, and this is called hemoglobinuria. Hemoglobinuria, and that's because you're seeing an excessive amount of red blood cells being broken down. And part of it has to do with this, because we have our urobilins. Remember that we all know what the color of feces is. It's going to run typically a brown, yellowish brown color, and this is what's responsible for it. So if you're seeing it in urine, it means that urobilins have made their way into circulation, and so it can be an indication of a high amount of red blood cell death. And this could be a lot of underlying reasons for this. Bilirubin, in some individuals, you'll see that if they drink excessively or if they have, just in general, if you see a yellow skin pigmentation, this is referred to as jaundice and can be an indication of liver problems, because recall that bilirubin is carried by albumin into the liver. And so if the liver is not functioning or processing bilirubin correctly, then you actually will see an accumulation in the blood vessels and it will cause a yellow color. So typically yellow skin pigmentation tends to be an issue with the liver, and it could be an indication of something very, very serious. And don't forget, in the large intestine, bacteria convert our bilirubin into urobilins and stercobilins. And lastly, the kidneys can get rid of any of these urobilins that enter circulation. And if you encounter intact red blood cells, that means that you have something we refer to as hematuria. Now, let's discuss that very quickly. There is absolutely no way these red blood cells themselves, let's go back to this picture. These red blood cells themselves can be secreted out of the kidneys. I know that at this very point in time, we don't have that common knowledge. These red blood cells could never travel through this pathway as urine. Absolutely never. So we know typically that if we're seeing intact red blood cells, it is because it is damage in the urinary system or the urethra. And let's look something up for that. Yes, of course, show all PDFs. And nope. 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 Here we go. And so, for example, if we look at our kidneys here, our kidneys are nothing but these filtering units. And as red blood cells, they flow through here. They just get skipped along. They move through it. There's no way they can actually be filtered through the kidney to be processed and then pass out along to our urinary bladder where we store urine. It's just not possible for red blood cells to go through there. So when we refer to this concept, and let's go quickly back here, hematuria, this means that there's a damage along the urinary tract. And what that is that once we have urine here, the only thing that exits out from the male system here is going to be urine. From the female system, only thing that exits out of here is urine. So if you're seeing intact red blood cells at this level, it's because there's damage along the exit pathway for urine. In males, that's the urethra. And in females, good news, it's also called the urethra. So I just want to provide some context for it. And so hematuria, the only time you encounter intact red blood cells. Otherwise, the kidney gets rid of components, right, of recycled or destroyed red blood cells, but never fully intact red blood cells. And so this is an indication of damage along the urinary tract. It has nothing to do with blood circulation. All right, let's close this. We won't need this. We won't need this one. All right. And now, so for this next segment, we're going to understand what is the difference, or what is type A blood, what is type B, what is type A, B, and O. And this table summarizes everything you really need to know. When you look at any individual, you have to imagine that if you're type A, an individual, their blood sample resembles this. If you're type B, it resembles this. AB is a blood type, it resembles this. Type O, it resembles this. Now, what is the difference? When we refer to type A, type B, if you look at the receptors of these red blood cells, there are surface receptors. Remember that we learned about receptors on the surface of cells like glycocalyx, right? We learned about those receptors that create a little profile or like little recognition sites on the plasma membrane of cells. And so if you're type A, you have a particular molecule that just has a different conformation, has a certain type of chemical structure that makes it different from type B. And if you've ever taken biochemistry, and one day some of you might, what you'll notice about molecules themselves is that it's just complicated Legos. That's it. It's like just having different atoms makes things work in different shapes. And so that's a simple way of really understanding amino acids and biochemistry in general as you move forward in your careers. But simply speaking, their chemical structure is different. So type A has this type B has this type. Now, we explore type AB, they have both. I think I'm confusing. This type B and type A, hence AB. So they have both of these surface receptors on them. And type O has none. It has no surface receptors. And so now what's the next part? That's the first half. You understood me so far? You already know half of the material here. In the plasma, antibodies, globulins, remember globulins were a component of plasma. Let's connect it with our previous stuff. Globulins came in two forms. We had our transport globulins. Right now we just learned about some of them. A transport globulin that carries iron. What was that one called? Starts with a T. Transferrin. And which one carries bilirubin, to the liver? Albumin, right? That's maybe not the same type, actually. But transferrin would be an example of one that carries ion. So let's write that in there if you have these notes. Transferrin. And albumin is its own monster. Albumin is a very complicated protein. It's worth sort of separating out. But transport proteins, transferrin is an example because it carries iron. Fe2+. Carries iron. And so globulins came in those two forms. We saw one right now, transferrin, in that previous recycling story. Now the main player here is immunoglobulins or antibodies. And so these antibodies are an example of globulins. And so they're swimming and they're involved in recognizing foreign things. Now let's think about the naming system because this is the only confusing part. Anti-B means that anybody who has blood type A has these surface receptors called antigen A. Antigen A. If you are type B, you have antigen B. Now, alongside these cells, if you are type A, in your plasma, you will have anti-B in there. And so this is telling you sort of what it's seeking. So what do we think what receptors is this one looking for? It's looking for B. And so try to remember as well that this binding is very dangerous. This binding, whenever antibodies bind to anything, it's usually telling the body this is something that needs to be destroyed. So if you have type A, antigen A receptors on your blood cells, you got nothing to worry about because your antibody B receptors will never bind to your own red blood cells. The danger lies in if you get, like someone's going to donate blood to you and they donate type B into your body system and it goes into your circulation, these antibodies will bind to that foreign blood, to that blood that it actually does recognize. And then it'll cause something we refer to as agglutination. So here's an example. If you put antigen B and then it gets placed into the wrong patient, you put it into a type A person. This person here is type A blood. Because again, if we look at this profile, type A blood means that they have antigen A, but they have antibody B. So that means that they have this antibody. And so if you donate the wrong blood type, which in this case we're seeing these are red or antigen B, or this person is type B blood. If you place it in, what's going to happen is that those antibodies are going to bind to them and then trigger the body to destroy the red blood cells. And not just that, they also just kind of clot, and then they clot in your red blood cells, and that can cause an instant it just stops your heart in general because there's no circulation, or it just triggers those cells to be destroyed. And so this is why it's important to type people before you donate blood. Now, let's finish this complete thought, and I hope it'll help understand the rest of it. Now, if you are type B, again, you have antigen B, and you have antibody A, and you don't have to worry about them binding. You're safe. Now, what is a person with type AB? They possess A, and they possess antigen A and B. So they don't have any of these antibodies. They don't have this one because if they did, it would bind to A, and then you'll have agglutination, which is a problem. If you have this one here, and this in this proximity will bind to the A receptors. I think I mixed them up. Oh no, the B receptors, and then it'll also cause this same process, this binding and hemolysis or destruction of those red blood cells. And so a person with AB cannot receive blood. Oh, pardon. Let's go slow. Let's keep going with type O. And now type O, the difference is that they have antigens missing. They don't have A or B, but they have antibodies A and B. So they're safe because it's not going to bind to any of those antibodies. Now let's talk about who can donate to who. If you have antibody B, you'll bind to what antigen? B. If you have antibody B, like anti-B, it binds to B. So if I donate this to this person, would there be a problem? If I donate this, would it be a problem? Why this one if I donate it? Why would it be a problem? Right, because it also has that B receptor. So this B receptor would cause a problem if I donate blood to this person with AB. What about if I donate type O blood? Would there be a binding to this? No. So now you might be able to understand why type O is considered, well, have you ever heard that word? What is it called? A universal donor, because their red blood cells won't trigger a reaction with anybody's antibodies at all. So their blood is very highly valued because it'll be donated to, you can donate it to anybody. Now there is one more called the universal recipient, meaning that they can receive blood from everyone because there won't be a problem. We can figure it out systematically. Obviously, type O, if we donate it, will it be a problem with these antigens? Antibodies? Will it be a problem if I donate this one that's missing any antigens, it's missing A and B, and would it be able to bind with any of these antibodies? What antibodies are here? None. So would there be a problem? No. So AB can receive from type O. Now, if I donate AB to type O, would there be a problem? Oh, yeah, there would be a problem, right? What about if I donated type B to type O? There's a problem. Lastly, type A to type O. Yes, very easy to understand, right? Now, let's finish up with AB. If I donate type B, would there be a problem to donate these to this person? What about this one? Would there be a problem to donate here? So type AB becomes our universal recipient. I'm just going to write UR and UD for universal donor. And who can type B receive blood from? There's only two individuals they can receive it from. Type O is the obvious one, type O. And what's the next one? Type B, nice and simple, right? So hopefully we all understand typing and why it's important before you get blood tested as to why it's important to know your blood type. And just as a little fun fact too, a lot of major populations, if you look at like who has what blood type, Native American populations in South America and Native American populations that did exist in the U.S., they're all highly type O, which is a very strange thing. Is that Native American populations in the Americas run really the higher percentage are type O. Everyone else sort of shares type AB and A, B, and B, A and B. And I don't remember, there's one more difference between some races, and I'll bring that chart up next time. It's pretty cool just to look at. It'll be fun to look at. And other than that, now, terminologies to things. It's always sort of a tough word to teach, but I think it's worth trying in anatomy. And the word antigens. You have to think to yourself that the word antigen can be absolutely anything. Antigen can be absolutely anything. It could be pollen. It can be a poison. It could be absolutely anything that binds to an antibodies. Antibodies don't change. Antibodies will always be these little globulin proteins that are swimming around in the plasma, and they're seeking foreign things. So the word antibody is always shared with the word antigen. Let's take this down. Antibody always bind to an antigen. And an antigen can be anything. Anything. In this case, it's these receptors. But if it was a little bacteria came, infected your body, a little virus infected your body, the antibodies, you will have little antibodies swimming around in your plasma, making sure to bind to them and destroy them. So you can see how viruses can be antigens. Pollen can be antigens. Anything that binds to antibodies is called an antigen. And so antigens aren't just these receptors. It's just something that binds to antibodies. And when you enter immunology, it'll matter. It'll matter. And so every single cell can be considered to have surface antigens because in some instances your antibodies can bind to them. Anything else I'm missing? And another word for these antigens, agglutinogens. Agglutinogens. Again, once more here. Agglutinogens. Here's our agglutinins. These are going to be the antibodies. Okay, so everything that we have here, right, this segment, let me just write it out. Dang it. Oh, man. Why is my pen so small? Okay, let's see. Glue. So the agglutinogens are found on the red blood cells, and the glutenins are the antibodies. And this process of where they react with each other is called agglutination. Up at the very top, it has it as cross reaction. And again, this cross reaction just forms huge clumps of red blood cells that can block passageways and cause death because, again, it'll stop the flow of blood in your body. This process, this cross reaction, is called agglutination. I'm going to write that down here. And then the last thing to add, you may have heard of blood types. Like there's two blood types for O. What are they? What's the two blood types for O? Positive and negative, right? You just seen O positive and O negative. That's just something, that positive and that negative comes from another surface antigen. That's also found here. All of these cells have the possibility of having, let me write it in. They either have the RH, actually I'm going to draw with the RH. It's a little bit easier to understand. The RH, they either have an RH molecule on them, or you have a red blood cell that doesn't. If you have a red blood cell that doesn't, it's considered negative. If it has that RH, it's considered positive. So it's a little bit easier on type O. So if you're O positive, that means that you also, on top of having the two antibodies in your bloodstream and having no A and no B, but if you're O positive, that means that you do have something we call RH. If you are, let's do the easier ones here, type B and we do RH, you also have B and RH. What's your blood type? B positive. If you don't have that RH antigen, then you are considered B negative. B negative. So this is considered from rhesus. It's called rhesus because it was discovered in rhesus monkeys. And all of these antigens, although right now in the greater context of things, or I guess there's enough material to really limit it here, is, you know, your body is always making sure that there's just nothing foreign in your system. And so all of these surface receptors serve to make sure that it's all just the same blood cells. Everything is just being monitored throughout in your plasma, or all these red blood cells are being monitored at any given moment. And that's all there really is to this last set. All right. Let's go to our new packet.